How many of you understand the process of new drug discovery? What many of you may not know is how long this process actually takes. With new and emerging diseases, time is critical. Not only does it mean money, more importantly, it means lives. The current average time from compound discovery to market is more than 10 years. Think about that. For a new disease that is discovered today, a drug treatment wouldn't be available until at least 2030. Often, a significant amount of time is spent working out the molecular target, which is the part of the cell that the drug interacts with. Over half of all medicines target a class of proteins called membrane proteins. It is therefore paramount that we develop rapid solutions to determine the structure of these proteins. Recent advances in the field of single-particle cryo-electron microscopy have allowed researchers to solve the structure of membrane proteins by capturing images of individual molecules at near-atomic resolution. However, solving the structure of a membrane protein with this method is still a monumental task, and the analysis workflow suffers from significant bottlenecks that result in these projects spanning years. One major bottleneck largely comes from the overall poor ability to automatically identify or pick particles within a larger image. There are a variety of auto-picking methods, including template-based, fully automated, and even deep learning, but all suffer from the same issue that is massively overestimating the number of particles in an image and creating a very large number of false positive images that must then be filtered out of the dataset. Filtering these data with current methods involves a Bayesian classifier, but due to all the false positive images created by the autopicker, this filtering step takes weeks to months and comes at a significant computational cost. Rather than another attempt at creating an autopicker, we wanted to make a tool that manages the output of all the existing autopickers in a better way, thus making it vastly more effective while reducing the time and computational costs associated with existing methods. CryoFilter efficiently removes the false positive images from the dataset. This translates to faster 3D models and more membrane protein structures. With more known structures, we can not only expedite drug development, but also design drugs that will have a higher chance of passing clinical trials, ultimately bringing safer and more effective treatments to those who need it most faster. CryoFilter employs a deep convolutional neural network that is trained to differentiate between good and bad particles on a level that current methods cannot match. This task is exceptionally difficult due to the overall low signal-to-noise ratio in these images. By sorting these false positive particles out of the dataset early, CryoFilter circumvents weeks of initial 2D classification and manual particle filtering and allows researchers to move further in their projects sooner. The network architecture we use is a customized deep convolutional network. It has two branches which learn discriminative features of the downscaled frequency-filtered particles from Gaussian-smoothed images and from images with Robert's edge detector applied. As a researcher in this field, I have seen the challenges with these workflows firsthand. Current solutions only address part of the problem, so developing a method that enabled us to combine existing solutions into a more streamlined approach was critical. Over the course of this health hack, we have successfully demonstrated that a neural network outperforms existing approaches in identifying false positive particles from single molecule cryo-EM images. This is an invaluable tool that will save structural biology researchers countless hours in the future. Now that we have answered our primary question, it's time to move CryoFilter into the next stage of development. Moving forward, we will look to do a direct comparison between CryoFilter and the current Bayesian pipeline in terms of processing time and true positive particle accuracy on larger datasets. And because our solution is open source, this tool can already be applied and built upon by structural biologists in labs around the world. These are the tools that we used when building CryoFilter. Of course, none of this would have been possible without the sponsors for this event, so I would like to profusely thank the sponsors for making CryoFilter possible. They have helped create a platform that will save countless hours of medical researchers' time and potentially countless lives one day in the future. And with that, I'd like to open the floor to questions.
Hi, Gavin. Thanks to CryoFilter team for a very impressive PPT for someone who's not a lab technician.
Gavin.
So,
Oh, sorry.
Oh, go on. I was just going to ask, this solution seems to rely very heavily on well-resourced and well-funded laboratories and technology and equipment. And I'm just wondering how your team sees overcoming and applying this solution in less resourced communities or countries or health clinics.
Yeah, make sure I'm not muted. Yeah, no. It's an excellent question. So it does sort of rely a little bit on that. Sort of the baseline of entry, the cost of entry is that you're doing single part cryo-EM, which is getting more accessible, but still at this stage is still quite expensive. I don't know if I have a very good answer for that at the moment, just that things are getting more accessible. We just built a new facility here at UQ, and I think as that becomes standard now, we build new facilities. For example, like I think Griffith might be one of the universities that we'd be looking at buying one of our older microscopes that now we can sell at a lower price and get more people in that entry level so they can start generating some data and using this tool.
Hey, Gavin, can I ask you, so I think this is really, really cool. And I just wanted to clarify what you focused on during the hackathon, because it's obviously this is a huge research project in of its own self. But if I understood you correctly, did you say that you trained and tested the CNN during the hackathon? So can you tell me a bit about your test data that you used?
Yeah. So that was the main focus because, like you said, this could be a massive project. So I went through ahead of time and I downloaded an open source data set. So this was of an ion channel TRPV1, fairly well known channel. It was a data set captured a couple years ago at the University of California in San Francisco. And one thing that's really nice about it is that it's a small membrane protein. And a lot of people that have taken approaches to try to apply deep learning to EM analysis use sort of these cookie cutter proteins like apoferritin that are like 2 megadaltons. I mean, massive cookie cutter proteins that behave very nicely. And then they build this nice network, and then someone goes to apply it to their actual research project, and it doesn't work anymore. And so that was specifically why I chose that. So the data I did, so I took that open source data, and then I went through and manually labeled 1,000 good side view particles, 1,000 good top view particles, and 1,000 examples of bad particles or false positives that were in the data set. And that's what we used to build and train the network.
Cool, thank you.
Hey, Gavin. I'm just looking at this in terms of the commercial gap, right? So you talked about that at the beginning that it takes all this time, around 10 years to deliver that drug. I'm just looking at how much time, do you have a view for us how much money you could be saving around this? What's the kind of impacts to research?
So the impact for research is quite large. I mean, personally, from some PhD student, I solved a structure of a human membrane protein for my PhD thesis, and the processing that data that I collected in 3 days took about a year. And I probably spent 2 months straight going through this exact process that CryoFilter is trying to circumvent. So just in terms of resources, in terms of scientists' time, that's one application. Another application would be time on the supercomputer. So for example, all of that 2 months is essentially time where I'm hogging 3 or 4 nodes on UQ's HPC system to process against this data. So there's 2 components there. I can't give you an exact number, but yeah.
Talking lots of money.
Yeah.
Yeah. Okay. No, that's cool. No, thanks, mate.
Gavin, it's Nina again. Considering the work that your HealthHack team have created just during this hackathon, moving forward, do you have the technical backing in order to make this actually happen?
Yeah, great question. So I'm not a deep learning engineer. I'm a structural biology PhD student, background in neuroscience. I'm okay with Python. One of the team members, actually, so I've spoken to the entire team. They've all agreed that we should get together after this and talk about the next steps and that they're comfortable walking through it and setting things up so that I can continue. One of the team members is in a lab just upstairs from mine, and we actually implemented CryoFilter onto HPC Wiener at UQ already. So it's already in a state where I could start troubleshooting that, and I do intend to continue working on it. Yeah.